Welcome everybody. We chose a topic today that we thought some of you might be interested in, and so clearly we've generated some interest around aging, which will affect us all. My name is Deepak Srivastava. For those of you who don't know me as yet, I'm the president here at the Gladstone Institutes, and it's my pleasure to welcome you all to Gladstone. I see a number of familiar faces and a number of new faces. We do this symposium twice a year for folks in our family, and so it's a pleasure to have you all here. We typically pick a topical area and dive into that in a way that we think might be interesting to you all. And we're grateful for the President's Council at Gladstone that supports this and sponsors this. And Will Evers, one of our co-chairs, is here today. Laura Dorman and Fred Dory. So thank you all for leading this, the President's Council, and many of you here are on our President's Council and on our foundation board. I want to take a moment to recognize and welcome back an important member of our family. Sue Desmond-Hellman and Nick Hellman are moving back to the Bay Area. They've been involved with Gladstone for a long, long time. Sue was the former chancellor at UCSF and is heading the Gates Foundation, and Nick is an infectious disease physician who's been very involved with this. So welcome back to your home. Now, since some of you are new here, before we go into our program, I just want to say a few words about Gladstone. We are a pure biomedical research institute. And we don't do everything, but we focus on some of the most major diseases of humankind that have been unsolved. And those are around heart disease, still the #1 killer, neurological disorders. We'll talk a bit about Alzheimer's and other diseases, neurological diseases that are really affected by aging, and viral and immune disorders, which you all have been hearing about coronavirus the last few weeks. And if you haven't yet seen, we did a wonderful public briefing here in the auditorium last week. It's online, and I encourage you to take a look at that if you want to get the real facts on this pandemic that is emerging across the world. So we focus on those diseases, but what we do that's quite unique is we're deeply integrating technology and data science into how we tackle those problems. And that is becoming more important now than ever in making progress. And we've hit an inflection point in the life sciences right now where things are accelerating at an unprecedented pace. It's really driven by technology advancements, and the data that are coming out of those technological advancements are really what's finally providing us insight about human biology in ways we never could even imagine before. And at Gladstone, we are never satisfied with just making those discoveries about how the human body works and how it goes wrong in disease, but we're committed to seeing those discoveries that might have a chance see the light of day in getting into patients. While we remain in our sweet spot of discovery science, we are very proactive in doing creative partnerships to get our discoveries into people. And it's our independent structure that really has facilitated that. So we're very closely affiliated with UCSF across the street, which is a wonderful institution that we're seamlessly integrated with scientifically. But financially and administratively, we actually are completely separate. And it's that independence that has allowed us to control our destiny in various ways and be creative in taking these discoveries forward. And we will continue to do that. And with the help of many of you in the room, we are doing that quite aggressively. So now to today's topic. I mentioned the areas where we focus. Each of those areas are affected by aging. And so they really could be considered diseases of aging. Over the next 40 years, in America alone, we are going to double the number of people from 50 million to 100 million who are over the age of 65. Double. Now, when you think about that, what's going to come with, and that's not even talking about in Japan, China, many other countries, those numbers are even more striking. So what's going to happen as that world emerges? Alzheimer's disease is going to go in the United States from nearly 6 million to 18 million over the next 30 years. Heart disease remains the #1 killer, as I mentioned. That is going to continue to increase, and with the obesity epidemic that we face, you can expect those numbers that have come down to be going back up for heart disease. And then when you hear about coronavirus, other viral infections, you hear about the elderly that are most susceptible to actually having the mortality associated with those, with the flu even. And why is that? It's because our immune systems also age. And you'll hear a bit about that today as well, how aging is affecting how we respond to pathogens as well. So in all those areas, we must do better. And at Gladstone, we're committed to that. And I think today's program will highlight some of the ways we're going about addressing the aging issue in each of those areas, and also considering how to rejuvenate and regenerate some of the damage that might occur over a lifetime, so that ultimately we improve not just lifespan of humanity, but health span. And that is what we think is most critical. It's not good enough just to live longer. We've got to live longer well. So with that, let me invite our panelists to come up for a discussion, and I'll briefly introduce them and have them say a few more words about themselves. So Steve Finkbeiner is one of our extremely creative scientists here at Gladstone, and is a neurologist by training, and will share with us some work that he's been doing and how he's thinking about neurodegenerative diseases. Casey Gifford is one of our star trainees who did her PhD at Harvard and came here for her postdoctoral training, and is going to be starting her own laboratory this summer, and is really got a superstar written all over her. And Todd McDevitt is a bioengineer who is doing beautiful work that he'll share with you on trying to understand how we can really engineer systems, and particularly in this case around secreted factors that affect aging. So with that, maybe if we go down the road, the way we're going to do this is each of them are going to say a bit about about themselves and the work they're doing and what they think is most important around aging. We'll have a bit of a period with some questions I'll discuss with them, and then we'll open up for some questions from you all and have a sort of free-flowing discussion. So, Steve?
Yeah, thank you. So my name's Steve Finkbeiner. Thank you,
here you
first of
go.
all, for coming. It's really a thrill for us to have you here, and I really hope you enjoy the time together. My name's Steve Finkbeiner. I left a tenure-track position at Harvard to come here to Gladstone 20 years ago, because I felt like this was really the place to be if I really wanted to make a dent in some of the disease-related science that we hope to do. In particular, being able, as Deepak mentioned, to move things out of the lab to really make a difference for patients. That's a real motivation for me. I'm a clinically trained neurologist as well as a neuroscientist. So I am a director of the Center for Systems and Therapeutics. I also direct the Toby Koret Center for Neurodegenerative Disease Research. Rick Meyerson's here tonight, representing Toby Philanthropies. And actually, you know, they had a big part in the interest I developed in aging. So I come from the neurodegenerative disease side, very interested in Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, ALS, Lou Gehrig's disease, Huntington's disease, frontotemporal dementia. And the thing that these diseases all have in common is the #1 risk factor is aging. And it's been really disappointing how often we've been trying to do clinical trials to develop therapies for these diseases, and they've gone on to fail. And so one of the things that we've developed in the Toby Koret Center is this common thread strategy where we look for things that are really cross-cutting across diseases that might give us a clue about targets that we could pursue that might have a better chance of being successful if we develop something to go into clinical trials. And boy, you couldn't pick a bigger common thread than aging. It really is the major risk factor, as Deepak alluded to, the number of Alzheimer's disease patients that we expect to increase purely based on demographics. And so I think for a long time, I'd say the interest in aging was mixed because a lot of people thought it was a sort of inevitable thing, that there really wasn't something you could work on or do much about. But that's really changed in the last few years. I think people have begun to discover important pathways that are involved in aging. And I think for me personally, one of the really landmark discoveries was induced pluripotent stem cells, because recently it's been possible to take a skin sample. I could take a skin sample of Gary, for example, and be able to measure a handful of marks, so-called epigenetic marks, methylation on specific proteins in your DNA, and I would be able to tell you within a few months how old you are. It's remarkable. It's called an epigenetic clock. We don't fully understand it, but there seems to be something about aging that leads these indelible marks that we can measure. And when we take cells from that same individual, reprogram them to be stem cells, and then measure the clock again, it's as if we've turned the clock back to zero, and it's as if those were newborn cells. And I think it's more than just a marker, because when we measure the physiology of those cells, how well they're functioning, they look much better, and particularly around pathways that are important for neurodegenerative disease. So they have really substantial improvements in the way that they handle some of the proteins that cause disease. And so we think if we could understand those mechanisms and processes better that control aging, it might really give us clues about ways that we can target that system in a therapeutic way to prevent or treat neurodegenerative disease, and for that matter, broadly age-related diseases. And so one of the strategies that we've taken, we began a collaboration with Google 7 years ago that really has helped us develop artificial intelligence and deep learning methods in the lab. And we're trying to train deep learning methods, deep network methods, to be able to tell how old a cell is, or how old the patient is from whom the cell came in a dish, and then to be able to use that as the basis for being able to do screens that can find some of these mechanisms for ways to target them. So that's just one example of sort of how we're approaching aging and how we think that that might lead to new approaches for rejuvenation and for treating neurodegenerative disease.
Thank you, Steve. Casey?
Sure. So I also want to say thank you all for coming. So I personally am broadly interested in the molecular mechanisms that control cell fate and function. So how does a liver cell know how to behave like a liver cell versus a cell you might find in the heart? But I was actually drawn to the Gladstone for something that Deepak touched on, which was to sort of turn those basic mechanistic understandings into something that's relevant for disease. And so as a trainee in Deepak's lab, I've been very interested in cardiovascular function and cardiovascular disease. And so my work often begins with a genetic association. And so there's a symptom or a phenotype, and we have looked at someone's genome, and we've decided we think that this mutation or part of their genome is involved in their symptom. And then my goal is to use advances in Cas9 as well as human-induced pluripotent stem cell technology to also, in mice as well, to then try and see if we can use these mutations to replicate the phenotypes we see in humans. And so we also are taking advantages of machine learning approaches and improvements in machine learning techniques, as Steve mentioned, to try and better understand all of these patterns that are involved in the phenotypes we're looking at. We're often using gene expression as our measurement, so they're very complex patterns that are defining these symptoms. And I thought I would maybe mention two vignettes that are related to tonight's topic of two projects that have been ongoing in the lab. And the first is related to calcific aortic valve disease. As Deepak mentioned, heart disease is the #1 killer in the country. Calcific aortic valve disease in particular is the third most common form of heart disease, and it involves dysfunction of the aortic valve. And so we actually undertook a small molecule screening project in human iPS cells recently with the goal of trying to identify a small molecule that could potentially serve as a therapeutic to either treat or prevent calcific aortic valve disease. And this is important because currently the only therapeutic option in this disease is surgical replacement. There's no therapeutic option. And so what's really exciting to us is through the use of human-induced pluripotent stem cells, and now with some follow-up work in mice, we feel like we've been able to identify a single therapeutic candidate that may, in the long term, may serve as an option for those that have aortic valve disease. And I think that's a really good example of using human-induced pluripotent stem cells to really understand the mechanism behind a disease and then also, bring it forward and try and identify a therapeutic. And then I thought the second vignette I would share with you actually is related to human-induced pluripotent stem cells on a very basic level. So we sort of piggybacked off of this idea that you could take a cell type, a somatic cell type that's completely differentiated, and turn it into something else that's functional. And so we, as well as many others, have decided that you could take fibroblasts and develop ways or experimental mechanisms or approaches to turn them into something functional, for example, a cardiomyocyte. And so this is important in the case of, for example, a myocardial infarction or a heart attack, where after the heart attack, your heart is full of fibroblasts, it's full of scar tissue. And so what this approach allows us to do is turn these fibroblasts into a functional cardiomyocyte. And that's important because we've shown in various animal models that you can use this approach to recover some level of heart function in those animals. And so that might actually be a therapeutic option in the future for those that are suffering for any sort of cardiac dysfunction. And so I think those are two sort of broad areas where we've been really trying to understand the mechanisms behind disease and really try to inform and develop therapeutic options going forward for these aging-related symptoms.
Thank you, Casey. Todd?
Great. Thanks. And yeah, it is a great opportunity. I love these kind of venues where we can just really talk somewhat informally and share what we're doing. And as Deepak said, my primary background in my lab, most of our focus is how can we use stem cells? And again, we're big users of iPS. But even before iPS existed, we were working with mouse embryonic stem cells to try and look at early principles of how can these cells turn into tissues? And that was our main question. Brief lab tour earlier got to show a couple of examples in our lab right now, things we're trying to grow and characterize for different modeling purposes. But early in the process, almost 15 years ago, early in the lab's history, we also realized that we didn't really even know what the cells were making. And so as we were trying to steer and direct the cells, we didn't understand why sometimes it worked well and other times it didn't. We started to realize we were never really asking or looking at what they were producing. And what they make can also sort of shape and direct what they become or what they can turn into. So we took our focus from also not just what can the cells turn into, but what are they making? And why that was relevant, or why it became really relevant for us, was a few years later, I was at a scientific conference, and this wasn't the first time, but it was one of the times I had seen this model being used by aging researchers in particular. And the term is heterochronic parabiosis, but what it means is they take a young mouse and an old mouse, and they sew them together, and they study the responses that occur in the old mouse and the young mouse. And so I was sitting there with my engineering or more translational hat on thinking, well, that's really cool science, and they're uncovering new mechanisms, and they're finding these. But what is the direct therapy from that? And that's not a very translatable path if we think of humans. So if it works and we get to a point, what could we do? Is there a substitute? And so actually turned our attention back and said, well, the cells we're studying in the lab, these pluripotent stem cells, represent one of the earliest stages of development. Not exactly like the young mouse, but could they serve as a substitute? And so that started us on a pathway where we started to think of ways that we could culture the cells in high density and collect that conditioned media, that which they basically secrete molecules into, and start to study that. And we started to make some characterization, and it's really been increased in the last couple years since we moved to Gladstone, working with folks here that do proteomic mass spectrometry. So this is a way that you can characterize all kinds of proteins at once, a very powerful technology, not easy to do, and hence why it's very challenging. But that gave us some insights. Okay, now we had a better idea of what was in there. And then one of the questions was, well, does it matter? Is there any function to this? And so we had looked at different model systems and what should we try to test this on first. In the heterochronic studies, they've looked at lots of adult stem cells from in the brains, in the muscle, and different systems. And we decided to initially focus on the blood for a couple reasons that Deepak alluded to. Blood, again, is a tissue. It's actually the tissue that is shared by all of our other tissues, so there's a lot of crosstalk with that. And so we were interested in, studying the stem cells in the blood. And so we did this now through a collaboration with a great researcher at UCSF at the Parnassus campus, someone whose lab had been doing these conjoined mouse studies for several years, and he had made some really seminal findings and discoveries with regards to brain and cognition. And so working with him, it worked out great, and he's been a great collaborator. So we now take some of these blood stem cells out of mice to first test this idea. We can expose them to the media that we've collected from our pluripotent stem cells, and then we put them back into the mice to test this. The reason we do that is that the aged or the old hematopoietic stem cells tend to not self-renew. They don't grow quite as well, so we can measure that. They also exhibit a propensity to turn into more of what's called the myeloid lineage, and that means that you have less lymphocytes on average in an individual. And this is what actually helps to stave off infections and challenges to your immune system coming especially from the outside. So we did that. We also see that there's an improvement in those white blood cells or the lymphocytes in this assay. And then we also can see that they actually are sustained when we transplant them. So those were sort of 3 functional criteria that have given us the motivation to continue pursuing this. But long term, we think that this is a much broader sort of thing, but we really needed to start with a defined focus. And so we're looking at sort of where could we apply this. It has a lot of still basic questions. We don't know what the exact factors are, so that's going to take us some time to understand. But also doing this from a cell source gives us a great opportunity to also manipulate this system. So using CRISPR, we can actually customize what they are making, and that enables us to do 2 things. One, we might be able to iteratively improve this type of cocktail because there could be things in there that are also negative, so we have a means to remove them. But also when we want to test what's called proof of principle, or the mechanism of these, we can look specifically and remove factors selectively and then test the cocktail again until we identify those factors which seem to be most important for mitigating this type of response. So that's sort of where we are right now. It's a really great state, and I've had a couple of fantastic trainees that have really driven this forward. But as both of the other speakers have also said, it's really this merger of the technologies that makes this possible. Without that, we just wouldn't be able to really address these questions as well as we need to.
Great, thank you, Todd. So I think you can see in each of those areas, it may or may not have been obvious, to accomplish those advances, it took a team of people coming from different backgrounds to actually develop the insight that was necessary, because in today's era of science, no one lab will typically be able to have the full range of approaches and technology and capabilities to really go deep into understanding these mechanisms. And you can't treat human disease unless you understand the mechanism. So each of these areas is really focused exactly on that. So Steve, I think a lot of people wonder when we talk about neurodegenerative diseases, people are fine for decades, and they're going about their business, and as they get older, then these things arise. What is it that we know now what is happening, and why does aging affect those and make those neurons more vulnerable, and what can you do to try to figure that out?
Yeah, so I think maybe aging does 2 things. One is that it makes our cells less able to handle the day-to-day stresses that they experience. And what that can do over time is that can lead to cumulative damage. So the other role time plays is that over time, the damage that gets done each day can accumulate in these cells, and we can lose them. It is true that we apparently can live sometimes 5, 6 decades, 7 decades without any symptoms. I think that's mostly a testament, actually, to how resilient and how many coping mechanisms, actually, that our brain has. We're quite plastic in that regard. So it's possible for us to cope and compensate for that damage and loss for many years before eventually the last coping mechanism is lost and we start to experience symptoms. So I'd say that's the other point, is that with some of these diseases anyway, with Parkinson's and Alzheimer's in particular, where we know it a little bit better, it's pretty clear that the underlying disease has actually begun quite a bit earlier than patients express symptoms. And so that is one of the really important things, is we need to figure out ways to identify these diseases and intervene earlier so we can make a difference.
So maybe going a step further on that theme, so when you developed this really neat robotic microscope that allows you to track individual neurons that we can make from human induced pluripotent stem cells over time. So when you say that when a patient presents with the disease, they've probably had the disease brewing in their brain for 2 decades maybe. And so you can't go back in time and figure out what happened. So maybe it'd be interesting for the audience to hear how you're using your induced pluripotent stem cells that are become neurons and that you can track serially over time with your microscope to really figure that out.
Yeah, so what Deepak's talking about is we built these platforms. I think we're on our 8th generation robotic microscope now, but the key capability these things have is we've built them so that they can identify and track individual cells over time as often and for as long as we want. We've gone 6 months, we can go a year, as long as we need to be able to observe as some features of disease first appear, and then we can follow those cells as long as we need to understand how that relates to what ultimately happens to that cell. And we can use that ability to track disease and visualize it to be able to create a blueprint for how the disease works, oftentimes borrowing statistical tools that are used only in clinical trials, because now we can treat each cell as if it was a patient and begin to understand that blueprint of causes, and then also, importantly, to figure out ways to intervene. And that's really been important for us for identifying the most productive places to sort of poke and prod to get the fate to be different. And I'll just add one more thing. So anyone who's followed what we've talked about closely will say, wait a minute, you just told me that iPS cells have 0 age, and now you're using them to study a disease that doesn't show up until you're in the 6th or 7th decade of life. Well, in fact, we have a couple different ways to make a neuron. So one of them is from a stem cell. Another is directly from other cell types like skin cells or things like that. And the cool thing is that when you make them directly from skin cells, you can retain some of those marks of aging. So it gives us this really interesting window to be able to investigate directly what the role of that aging signature might be and how it impinges on some of these pathways that cause disease.
Great, thank you, Steve. So, Casey, we talked about this disease that occurs in the heart valves, and that gets worse as people get older and progressive, and then people need open heart surgery, ultimately to replace this valve. A similar process goes on in the vessels where they get hardened and calcium deposits. Many of you have probably gotten calcium scores done and to tell if you have hardening of your arteries. Liz Blackburn, who is one of our Nobel laureates here at UCSF across the street, had described the role of telomeres, which are caps on the end of chromosomes that get shorter and shorter as people get older, then as potentially something associated with aging. Casey, I was wondering, maybe you could share with folks how we're thinking about the telomere lengths and how it might impact human disease, in particular the model that we've been studying.
Yeah, and the ability to model disease, right? I mean, so we've mentioned a little bit how we often use mice to model disease. And it turns out mice have longer telomeres than humans. And we actually have had a hard time modeling certain diseases, especially cardiovascular diseases in mice. And so it turns out that one of those reasons might be that telomeres are longer in mice. And so what we've actually done is shown that if through genetic manipulation, if you shorten the telomeres of mice, you can then create a better genetic model in mice that replicates human symptoms a lot better. And so it just speaks to, you know, our ability to model diseases, and sort of this is one of those basic mechanisms that we don't quite understand telomere length and how directly it relates to aging, but I think it's in particular for the calcific aortic valve disease, it was very relevant, where as soon as we limited the length of these telomeres, we were able to replicate that phenotype, which had been completely masked previously. And so who knows what other human diseases we've tried to study in mice, we were also suffering from this problem. And so I think it'll be a really interesting point going forward.
And some of our folks in the neurodegenerative area have done a similar approach and are shortening the telomeres and are actually seeing acceleration of the mouse symptoms of some of the neurodegenerative diseases at an earlier age. And so I think this will be an interesting approach to understanding that. So Todd, this notion of something that is present in young people's blood, making an older person young again, is really science fiction-y. Yes. And if there are pictures that are almost disturbing, but you see two mice actually sewn together, and their blood intermixes, and then the old mice starts to become young again. It's really remarkable, so it suggests an important thing, and that is that with the same cells that one has as you're older, there is something that's reversible in those same cells. It doesn't have to require new cells going in that are younger. So how should we think about it? It's a really paradigm shift in how we think about aging fundamentally, I think.
Oh, absolutely. But take the most dramatic example, IPS example. So that is also an epigenetic, as Steve was saying, switch. Now, in that case, it's classically been done that you overexpress proteins that force the cells directly to that fate. But because it's epigenetic, it suggests that there are potential pathways that just are existing, but we just don't know how to access them. They're complex, probably multiple inputs, and that's probably the most dramatic example. I don't expect that if we took our adult cells, put them in this secreted kind of stuff, that they would go all the way back. But maybe there's enough signals that sort of remind the cell of when it was in a younger state, or it can restore some of the things that maybe it's the absence of something that it hasn't seen for a long time. And those signals for even a short period of time might be enough to sort of bring back that function. It probably goes to the fact that there's a lot of conservation of those underlying pathways in the cells, but what changes is a lot of the environment and the accumulation of the exposures to, or accumulation of mutations we get because of the stuff we get exposed to over our lifetime. So I think it is sort of suggests that there's a conservation of at least some level of this endogenous regenerative capacity. I don't think it's infinite by any means. I won't go say it, but it does suggest that there may be some things that can go back or at least be rejuvenated to some extent.
Yeah, I have a number of other questions I could be asked him, but I want to make sure that I leave time for the audience to have some questions. So let me open it up there, and if there, then I can come back to some of my questions, maybe. Yes.
Learn from other species that live a lot longer, like sharks that live 300 years? Are we looking at learning from those cells?
I mean, the short answer is yes. I think there are a lot of people that are looking at both animals that live a lot longer, but also shorter, and trying to understand. And I think telomeres is one example, that understanding telomere biology and how it compares between these animals is extremely useful. And so I mean, that's the short answer, is that absolutely there's a lot of work that being done, both academically, and then Google has an entire company called Calico, whose goal is to study aging, and how basically their goal is to make us live forever. And so they do a lot of studies with the naked mole rat, for example, which is an experimental model that's not very pretty, but it does live for a very long time. And so they're doing some really exciting work, both molecular work, genetic work, epigenetic work, to try and get at that question.
And I'll just add, you have to Google naked mole rat to get a good image, because
See how ugly it really
they're
is.
really hideous. Plus, they live underground, piled on each other, breathing each other's CO2. I'm hopeful that we can find another solution besides that.
I wouldn't pick them up. I don't.
But you may know that in terms of so many different species do have fundamentally different lifespans. So that means that the fact that humans could reasonably live 90 to 100 years isn't a de facto written rule. There's some reason for that. We do know that from beautiful work by Cynthia Kenyon and many others, that mutating a single gene in a worm can increase the lifespan by several fold. And so that says that it is clearly genetically controlled. And so one could, in theory, unlock that. Going back to what I mentioned earlier, our goal in our, at least, aging research is not necessarily to unlock that and increase our lifespan, but really focusing on health span. Obviously, we could talk about the societal consequences of being able to increase lifespan, which it would be significant from many perspectives.
Can I just add one thing? So I think there's also some really interesting research that I wanted to bring up that's been done in fruit flies, which shows that if you delay the onset of puberty, you actually delay the onset of aging. And so that's an example that's sort of both hormonally and perhaps genetically encoded that could eventually influence lifespan too.
I could have wanted to do that for my teenagers, but delay it permanently. Sharon?
I'm sure you're aware of research on autophagy in mice. I think it was the subject of the 2015 Nobel Prize, showing that induced autophagy through fasting in mice had significantly positive effects on neurodegenerative diseases and the removal of amyloid plaques and other harmful substances in the brain. What do you know about that? Is there any work on humans? Does the mouse work translate to humans? Instead of spending millions and millions trying to find a drug to cure these things, should we all like skip breakfast or what?
So we happen to have one of the world's expert on autophagy in neurons here, so Steve will address that.
Yeah, so exactly. So in fact, so for those who aren't aware, autophagy is one of the two major pathways in your cells that are involved with clearing damaged proteins and damaged organelles. It's also involved in certain infections, can be useful for clearing those. And it's gained a lot of attention in the neurodegenerative disease area initially because it was the only pathway that people thought might be capable of clearing those aggregated protein deposits like plaques and tangles that are commonly a feature of neurodegenerative disease. But as you point out, there have also been these tantalizing connections to diet and caloric restriction. In particular, there's a pathway, at least one of the two major pathways that activates autophagy is through caloric restriction. And it's believed that in addition to clearing garbage, the autophagy pathway also is a recycling pathway. So if cells are starved, what autophagy can do is chew up proteins and release them for reuse and generating energy for the cell. And so what people have found in a variety of model organisms particularly, so mice and flies and worms and other things that we often use in the lab, is that if you activate this pathway, you can increase the lifespan of the animals in which the activation occurs. The one caveat, I'd say the one open question between some of the studies in those short-lived organisms and humans is kind of to your question, how translatable are they? Is what we learn in a fruit fly, especially with regard to longevity, can we exactly cut and paste that for a human? And I think that's still an open question. It's not clear whether exactly the mechanisms of aging are going to translate in such a way that you could implement that. At the same time, I think I have a particular colleague in my field who's trying to do that personally. He looks really starved sometimes. I worry about him. But he's trying to change his own lifespan by implementing basically this very rigid fasting approach to basically stimulate autophagy to the extent that he can.
Yes, Fred.
Yeah, Alzheimer's has been almost the Bermuda Triangle for early stage companies and a number of drugs. What have we learned from those failures?
We've learned a lot about how to fail. Yeah, I mean, I think, is the question, So one major problem, I think, in that field, from my perspective, is that there was one idea about what caused Alzheimer's disease that really took hold years ago, and all the drug companies that have invested a lot of money and effort, you just saw another clinical trial failure today, in fact, that lowered stock on Lilly and Biogen, is really this amyloid hypothesis. So this is one idea of how Alzheimer's disease is caused, where it's those plaques that might be driving the disease. And honestly, from a scientific standpoint, I thought that the evidence that really showed that those were the pathogenic mechanism of Alzheimer's disease wasn't super strong. Even if you just considered the correlation between someone's clinical presentation and how much plaque they had in the brain, you can have plaque with relatively normal cognition, and you can have Alzheimer's disease without plaques. And so at that stage even, the link wasn't super direct. Nevertheless, there's been a huge investment, and that's been the major focus for most of the efforts, Biogen, Lilly, others, Roche, where they've especially been developing antibodies or small molecules, Merck, to try to block that process. And so that, so far, really hasn't worked. And I was just down in San Diego in a deep dive at the Adler Symposium on the Biogen trial and the Biogen data, which they're trying to resurrect for FDA approval. And it's really difficult to see that going forward. So I think that's a big reason is that drug companies placed all their bets on one idea, and that idea, so far at least, hasn't held up. Is it possible if you intervened early enough, you could find a time point where that would make a difference? Potentially. But a lot of people wonder if that's even feasible. Could you afford to do a clinical trial that would be that long? I think it's an open question. So I think we desperately need basically new approaches. One of the targets that's being worked on here at Gladstone is tau, and I think there's some really encouraging data in preclinical models, and I think a lot of hope that targeting tau might be useful for Alzheimer's as well as some other diseases. There are also other mechanisms. Neuroinflammation is a disease where if you look at just the genetics, boy, it really screams at you that the immune system is playing a really important role in Alzheimer's disease and also in other neurodegenerative diseases. And one of the challenges in science is that you tend to kind of be the drunk under the light post. If you're a neuroscientist, you look at neurons. You don't look at the immune system. But that's one of the nice things about being at Gladstone and being in such a collaborative environment is it really gives us an opportunity to sit next to a card-carrying immunologist and to be able to have a conversation and think about ways we can work together to tackle these.
And in fact, with the generous gift from Nick Simon, one of our trustees, we're just launching a new neuroinflammation center to get exactly at this area where we have strengths in immunology, strengths in neurology, and we want to have those converge more to really get at the basis of some of these diseases. Steve, it might be good to just comment also on how we're, given that we've been an epicenter of work with ApoE as a genetic contributor, to work on that in that space as well.
Yeah, thanks. I'm surprised Bob didn't throw something at me. I apologize. So if you look at the genetic signals, by far and away the biggest genetic contribution to Alzheimer's disease risk is this version of apolipoprotein E that you have. It comes in a couple different versions, and depending on the one you have, you're at substantially increased risk for Alzheimer's disease. And Bob has done, lifelong work. He's, a member of the National Academy based on the contributions that he's made, particularly in that area, and has really been a vanguard in terms of trying to think about how could you take advantage of that knowledge and translate it into a therapy that might make a difference for patients.
So I think the bottom line is a lot of approaches that are failing now the process got started maybe a decade ago even based on that knowledge. And I think will be an example of how our current knowledge base of mechanism is very different. And the things that will now begin to enter lead up to a clinical trial over the next decade, I think is where I have hope because it'll be founded on a much deeper mechanistic understanding. Yes, David.
Thank you. One of the major problems, obviously, is conducting clinical trials within a lifetime. And with these parameters that you are discovering and testing, how far away are you from having a surrogate marker that would allow you to do a clinical trial in 5 years with an endpoint that you could believe?
You're on again, Steve.
And, I always love the how far away questions are you. Although I will say, one of the best things about Shin Yamanaka, he was asked apparently by a news source like when you would ever be able to make a stem cell.
He's the one who invented induced pluripotent stem cells.
Yeah, so Shin is here. He got the Nobel Prize for it. I think he said something like 15 years he guessed, and then he discovered it 2 years later. So I do. So I think one great thing, exciting thing about science is you never know when you roll in the office that morning what new discoveries you'll find. But the question really is, you're absolutely right. I think there have been a lot of criticisms of the clinical trial designs, I just came from a meeting where we were picking over some of the debris from the Alzheimer's disease clinical trials and to think about how those could be done better. And so there is a huge effort to try to develop biomarkers, and I think the most promising ones right now are these things called PET ligands. And for those who aren't familiar, you're probably familiar with having an MRI taken. Now you can develop, they're almost like drugs. They're small molecules, but they're radioactive. You give them to patients, and they can go through your blood and actually stick to certain pathologies that are in your brain. It could be the plaques, it could be the tangles, different tau deposits, things like that. I think there's a lot of optimism around that's going to be very useful. And I saw some great tau PET data that's coming both from this Biogen trial at the end, but now is really being implemented in a lot of trials going forward that I think is very promising. There are some other biomarkers in blood and the CSF that also look really good, and I think together we can use those to make very solid diagnoses of Alzheimer's disease. But I think these biomarker data, particularly tau, is going to be very helpful. And for those who aren't familiar with the role biomarkers play, they're very helpful both in enrolling patients, because if you have a drug that treats tau tangles, you want to make sure your patient has tau tangles if you enroll them, because if they won't stand to benefit. And so that's one reason. But the more important one is that it gives you these early readouts of whether your therapy is having an effect on the target that you're trying to pursue. And unfortunately, the way a lot of clinical trials up until now have been performed is they haven't really used those biomarkers, and what we've used instead are clinical measures. I test your memory, or I test something else about your brain, and those measures are okay, but they're not great, they're not super quantitative, they don't change a lot. It takes a long period of time to really measure those changes. So all that noisy variability means that the clinical trial's not very sensitive, and it takes a long time to read out. That makes it extremely expensive for drug companies. And so in this kind of mantra of fail fast so you can succeed, I think having really good biomarkers could, it's a game changer. Multiple sclerosis is a great example. For those of you who don't know about that disease, it's a neuroinflammatory disease. When I began my residency, we used to give steroids to patients with multiple sclerosis. It was just awful. And now, due to MRI imaging, where you can see those abnormalities, it's completely changed that. And so now there are many drugs available. It's made huge progress just in my lifetime. Very exciting.
Great. Wonderful. You guys are right on time. It's 5:30 sharp. Well, please join me in thanking our panelists for stimulating discussion. Thank you.